It's crazy how like biblical miracles eventually become tech companies. We've released phase 3 clinical trial results in a 38-patient study in Europe showing the first time in the history of the world, as far as we know, that patients that can't recognize faces have been able to read again. In the Biohybrid neural interface work that we do, where what we have is an electronic device that we grow up in a dish with heavily engineered neurons seeded in it, we can optically stimulate and electrically record. When this is seeded into the brain, those cells grow in, wire up, form new connections into the brain. The vertical integration is essential. We do everything from integrated circuit design to MEMS fab to animal surgery. We have a large in-house animal program because that is where we can continue to learn things quickly. Like you can learn something every week in mice. You can learn something every 3 months in primates. You can learn one thing every 5 to 7 years in humans. Company's called Science. We're about 3.5 years old. I've spent most of my life interested in like engineering the brain broadly. I mean, my origin story, I saw The Matrix when I was a kid, and it's like, I don't know if we're living in a simulation, but we should definitely build one. And ever since then, that's kind of been the course I've been on. I talked my way into a lab doing neural recording in monkeys my freshman year of college, and that was really where most of my education happened. It's such a broad technology in that there's a poster in our office it just says alter the brain, alter reality. Like we're used to dealing with the world out there, engineering, building buildings, building rockets, building electronics, but really your entire conception of reality is rooted in your brain. And if that, and so there's this interesting inversion that's possible there. And it's both when you lose the function of your brain, this is a very profound loss. But this gives us an incredibly rich, I think, engineering substrate for things that are really going to define a big part of the future.
Yeah. So I want to get into the details of like what is neural engineering and like how does it happen? How are you guys doing it? What are the different things you built? But I think maybe the simplest way to anchor the conversation is just saying at the beginning that you've sort of like cured blindness in a way. Like
Yeah, well, I definitely wouldn't use the term cure blindness.
Fair
I mean,
enough.
we have an ambition to cure blindness.
Amazing. Well, tell us,
yeah.
what do you mean?
The lead product, the thing that's furthest along. So when we started the company, there were a couple of different ideas. The first was a very different approach to building neural interfaces, an idea that I had originally had back in college, but was really beyond my ability then and really beyond the ability of like other resources like collaborators and researchers that I knew, called a biohybrid neural interface. But that is a big research project. That is not going into humans yet. There's a ton of neuroscience to do there. There's a ton of cell biology to do there. And so we thought that is very difficult. It needs to be paired with a kind of more practical business, because at the end of the day, in order to spend these types of resources on this type of research, there needs to be something that we do that can realize value sooner. And it struck us, me and my co-founders, that the thing that would be most feasible with what the technology available circa 2021 was restoring vision into the optic nerve in patients that had gone blind because they had lost the light sensitive cells in their eye, the photoreceptors. And that seems to have been borne out. So over the last 3.5 years, we've developed some technology internally. We've also done some acquisitions, and long story short, recently released phase 3 clinical trial results in a 38-patient study in Europe showing the first time in the history of the world, as far as we know, that patients that can't recognize faces have been able to read again.
So unbelievable. I've seen the pictures of like, what does it look like before? And there's basically it's like a dark spot in the middle of your vision because your rods and cones have died.
Yeah,
That's
so only like the central 6 or 7 degrees of your field of view is high resolution color vision. All of the outer peripheral vision is lower resolution black and white. And the approximately, right? There's always this is biology. There's no like hard rules. And so that center pit, you probably know as the fovea, the area around the fovea is the macula. And so there's a disease known as macular degeneration, which is just like when the macula degenerates. This is where the big concentration of cones are. Often called age-related macular degeneration because there's a form of it which is strongly age correlated in the mid 70s. It becomes pretty common. And when age-related macular degeneration gets bad enough that it really like the whole central area of vision has degraded, this is called geographic atrophy because there's a geographic area of the eye that has atrophied. And when you have that, like that part of the visual field is gone. And if you lose that central visual field, that is the most important part of the visual field for doing things like recognizing faces, reading, interacting with the world. And so these patients still have some residual peripheral vision, but they've been unable to use like the most important parts of their vision. And so restoring that is very powerful, and then the brain fuses it with the peripheral vision, and so it fills in. And there's some information there that's not actually real, but you can give them, like if you show a patient like a line, um, the implant right now is only able to do black and white at very high contrast. And so if you give them like a green line, they'll say, "Oh, it's green," and then it turns white, and then it turns green again. But it appears as a solid percept because even though the implant doesn't actually fill the whole atrophy, the brain fuses it because the brain is always constantly filling in. Um, if you think about like the eye is not a camera. Um, the the brain has this strong model of reality. It has this internal world model, which is really only fairly weakly modulated by the senses coming in through the cranial and spinal nerves. And this is one of the reasons that illusions can be very strong. Um, and if you think about what is the image that actually falls on the photoreceptors, so first of all, the eye is backwards. The retina is kind of
'Cause
backwards in
light comes in
vertebrates.
and like yeah, yeah, it
Yeah,
inverts
for
when it hits you,
yeah, and
yeah.
this is for there's a couple of animals that this isn't true. It's kind of a quirk of evolutionary history that it worked out this way. This is also the reason why you have a blind spot, because there has to be if the most frontmost layer is the optic nerve, then there has to be a spot where it like dives and exits through where the photoreceptors would have been.
'Cause you can think about it like there's an image projecting on your eye, it, like, hits the back of your eye, and that's what's exciting all those cells, but there's gotta be somewhere where that gets out, and
Exactly.
that's the
And now,
optic nerve, and
if
that's where the blind
yeah,
spot
and so
is.
then the animals that where their photoreceptors are in the front layer and the optic nerve is in the back, there's no blind
Hmm.
spot,
Oh,
right?
smart.
Because
Yeah, it
then
makes
there's
sense.
no. But we kind of like evolution did this a long, long time ago. Most vertebrates have this, but so there's really three layers of cells in the eye. There's the photoreceptors. The first synapse past that is called the retinal bipolar cells. Bipolar because they've got two ends. They connect the photoreceptors to the retinal ganglion cells. And ganglion is just a fancy word for like neuron that goes somewhere else and connects somewhere. And the retinal ganglion cells are the optic nerve. Um, and so these patients, they've lost the photoreceptors, but the bipolar cells and the optic nerve is intact. The device stimulates the bipolar cells, which is the first cell layer past it's the first possible opportunity to get the visual signal back into the brain. But the kind of to go back a second, the light shines in through the eye, it filters through the retinal ganglion cells, through the bipolar cells, and then eventually gets to the photoreceptors. And so you've got like it's like a little blurred. You've got these like blood shadows of blood vessels over it. And so the eye, as it's moving around, is filling in this information. But the brain is just getting like the actual image, if you think of it that way, would look like pretty occluded and pretty blurry. But the brain has this model of the world. Like I hesitate to draw a analogy to something like Sora, but like you kind of can. And then it's just constantly getting this partial information from the environment that's updating that world model. And so when you give them this little bit of information, the brain will fill it in,
So
and you
yeah,
get
that was definitely a
yeah.
question I wanted to ask because the way that I understand it is that you can put a patch basically like a little would you call it a patch? I don't know what you call it, but you put something on the back of the eye to basically replace those cells, replace the rods and cones that are in the center area that you've lost. But wouldn't you need to cover a lot of the eye? This was what I was going to ask: is wouldn't you have to cover a lot of the eye to really regenerate a lot of vision? But it's actually just a spot, and then the eye does the brain does the rest.
Yeah, so in these macular degeneration, these geographic atrophy patients, they've lost anywhere between 2 to 5 square millimeters on like typically, sometimes a little more, of that central high acuity visual area. The implant right now is about 2 millimeters by 2 millimeters. And so it doesn't completely fill that area of atrophy, but it gets a bunch of it. And so when you think about increasing the visual field, the if you fill the fovea, as the electrodes or the pixels on the implant get smaller, you can convey more information simultaneously. So in kind of like at one glance, you can get more fine detail. You can have smaller characters that they could recognize, but it is still a constant section of the visual field in terms of like degrees. Um, the and the eye is constantly moving around and filling in information. So you don't notice the fall off in resolution. And so if you can get the fovea and you still have a lot of the peripheral vision and you can allow the eye to saccade around naturally, then the world model is going to get that information.
That's so cool. So I have so many questions that are like the details of this sensor and like how the information conveyed. Can you give us people a sense first though of just like the full end-to-end system? Because there's something that has to get that information from the world into the implant. That implant has to convey that information to
Yeah.
the other layers of cells somehow. Can you just give us like the sort of end-to-end how light from the world gets into my brain
Yeah, so
using
like
the
that whole signal chain.
basically?
The brain is an information processing organ. You can think of it in terms of information flows. So there's two parts. There's the implant, which is it's a photovoltaic array. So if you see a picture of the implant, there's all these little hex grids on it, and each one of those is basically a little solar panel. And so if you shine laser light onto the implant, if there's intensity, it's like if you think of it like an overhead projector and you are projecting onto a screen, there'll be some part that's dark, and then there's some part that is you've projected light to have a shape. Um, so wherever you're projecting like light intensity, it'll be absorbed by that pixel, and then that energy basically gets released to create an electric field directly above where it's been absorbed, because it has all these little solar cells. And so that is inserted under the retina, and then the patient wears glasses, and these glasses have a laser projector on them that is used to illuminate that implant. And so
There's a little camera that's looking out
yeah, so
and sees
the
the world.
glasses get a video feed from somewhere.
Yeah.
Now, in practice, this is usually a camera on the glasses, but it could be from anywhere. You could project like a virtual screen. You could connect it to a computer, but you need a video feed from somewhere. There's a camera on the glasses. That is what they usually use. Um, and then in the trial, there was no eye tracking, so the patients had to look straight ahead and beyond about 12 degrees. No, it was more than that. It was like beyond about 15 to 20 degrees, they'd have to move their head in order to see more of the page. The next version of the glasses have eye tracking on it so that they can move their eyes more naturalistically, and there's a wider field of view on the world camera so that you can figure out, so the glasses are getting a big field of view. You figure out where they're looking, you reproject that, and then you send it. And then that really allows the brain to fill in more of it much more
Was that a
naturalistically.
limitation of the cameras, you know, whatever angle, or was it more of about the laser hitting the
So
implant?
both.
Okay.
So we're working on a new version of the glasses right now with a bunch of key improvements. The first is there's a thing called the eye box. So like like if you look into a microscope, if you're lined up with the microscope perfectly, you get this big clear image. But if you get out of alignment with it, the whole thing vanishes. So that's called the eye box, which is how aligned do you need to be with the light in order to get the image, which is different from the field of view of the image. And the glasses that were used in the clinical trial only had an eye box of about a millimeter. And so the patients had to be lined up with it exactly. And
And
if they got it,
they go down your nose or something, then it's off and it's just not gonna work.
yeah. And also the laser source was this external fiber coupled box in their pocket, which would like get yanked on and it would get out of alignment. So when it was lined up, it was really powerful. It was a very clear proof of concept that you could restore vision this way, but it was finicky in a bunch of ways that have nothing to do with the implant or the neuroscience. And so one of the big things that we're doing now is we're increasing the eye box to hopefully as much as a centimeter, so that will both make it less finicky for the patients to wear, but also means that the eye can saccade around for a wide field of area of movement. You can couple that with eye tracking, and then you can do saccadic reprojection to allow them to use their eyes much more naturally. And this is no change to the implant, just to the glasses, which of course are easy to reissue to patients.
Very cool. So this, in my layman's interpretation of all this, this is sort of like replacing a rod more than a cone in that it's a black and white signal, in that it's probably a little bit lower resolution, that sort of thing. How hard is color? How hard
Yeah.
is
So,
a
and
cone?
the inventor of this technology, Professor Daniel Planker at Stanford, the way that he described this device when he was developing it a while ago, were electronic photoreceptors. And so that's definitely a totally reasonable way to think about it. The So color is interesting. The so there's so we're activating the bipolar cells, which aggregate up a bunch of rods and cones in kind of a very structural, in a very like precise way. And there we when we think that, if not we think the next version of the implant will give us a bunch of grayscale. and probably maybe some uncontrollable red and green percept. In another generation of the device, we're going to go from about 380 pixels to about 7,000 pixels in the same footprint. The current version of the implant, each one of those pixels is 100 microns across. Photoreceptors are much smaller than that, and bipolar cells are much smaller than that. You maybe call it 10 microns. And so you want to get to ideally kind of single cell resolution on that. But as the uh, hard problem here is that as your electrode gets smaller, when when you create this electric field, the depth that it penetrates into tissue gets shorter and shorter. And so you have to get more creative about, like, there's if the cells that you want to activate are some distance away, and you need to inject a certain amount of charge or create a certain field strength in order to drive them. Then as they get smaller, this gets harder to do, even though you want to get the spatial resolution of like a cell. Um, Now, there's some bipolar cells that if you excite them selectively, we know this will drive red. And there are some that if you excite them selectively, you'll get green. And those kind of aggregate up red and green uh, photoreceptors below them in this like opponency pattern, because it knows that when light is shined onto the eye, you will drive one but not the other. And so the bipolar cells kind of aggregate over this. And so it should be possible to selectively excite retinal bipolar cells to get red or green percepts. And as the electrodes get smaller and more selective, you should be able to do that more selectively. We think that the next version of the implant will probably get some red and green percepts, maybe not so perfectly controllably, but on that path. Blue is a little more challenging. It's not clear that blue is possible through pure excitation. I mean, you might have to inhibit some cells while you excite others to get blue. But this is still, I mean, there's on the one hand a lot that's known about this, and this is ongoing research, but there's many types of retinal bipolar cells. There's dozens of types of retinal ganglion cells, and it's still a very active area of ongoing research to really tease apart all of that physiology.
Wow. So is the challenge of getting to smaller and smaller sizes, like you talked a little bit about how, it's hard to specifically target a thing because you're within, yeah, it's like behind a lot of other stuff and whatever. But is it more of a laser problem right now, or is it more of a sensor problem in terms of shrinking down the actual
It's on
size?
the yeah, no, it's
Okay.
on the electrode side to shrink that. Yeah, I mean, you can confine the laser spot just fine. The And I mean, even we're not at the limits. We're not even at the point of things like adaptive optics. Like, there's all kinds of stuff that you could do on the projection side to get really small spot sizes on the retina. But the uh, you've got this like like this material that's absorbing the light, and then releasing that. And that you want this to be, first of all, not in the visible spectrum, because you don't want it to be in bandwidth. Like, you'll get some scattering, it'll interfere with other vision. So this is in the infrared. In fact, our peak is at 880 nanometers. And so you want a material that has a very high quantum efficiency for converting infrared light into these fields. And that we are pretty close to the limits of how efficient that can be. Um, and but you can do things like you can go 3D. Like, right now it's a purely planar collector and planar electrode, and you could have pillars, you can have other structures, you can have your return electrodes be several pixels away, you can move from a purely passive analog device to something where you use kind of the entire surface as a collector, and then you have internal electronics using more of a digital process um, to send that where you want it with higher energy densities. There's a huge design space there that we're at the beginning of exploring.
That's so the fascinating thing about that there's this big design space that and this big opportunity of like figuring these things out is like, why wasn't anybody else doing this? Like, why, like it seems like you probably found this thing that was sitting in academia for a while and it like was
Yeah.
possible, but no one was actually doing it.
Yeah.
Like, why
Well,
was nobody actually doing it?
also, how this came to us is kind of an interesting story.
Yeah.
So we did not pluck this out of academia. We got it a little bit out of obscurity, but we're actually the second corporate owners of this technology. So we acquired a company called Pixium, which was a publicly traded company in France, which ultimately ran into an object lesson about why it is difficult to be a publicly traded company in France. Um, and the but and for Pixium, it was actually their second retinal prosthesis. Um, and the why it ended up there is kind of a long story and kind of a quirk of history, but it wasn't. I don't know that I would say it was neglected. So Daniel Planker is a faculty member in the electrical engineering department at Stanford. This technology started probably around a decade ago with money, philanthropic money from, I mean, well-known Silicon Valley investors that cared about ophthalmology, and so it has always been. Um, and so it's tough to say that it was totally neglected, but yeah, I mean that's not. I mean, it's still. It's like why did we end up in this position now? Why was this not done a while ago? I don't know that I have
I
a
mean,
great
well,
answer for
then the
that.
follow-on question is like, what are the other things that are like that, right? I mean, if there's
Yeah,
this opportunity was just sitting there,
yeah.
and there are people that now have sight that didn't, and like,
What are
what are
other
the other
things like
things?
this?
What are the other?
Yeah, and I
There's
mean
got
the
to be tons
answer.
of
I
them.
mean, maybe a better way to answer this is not that this was like super special and that it was neglected and that we were such geniuses to find it. And we were paying attention. We knew a lot about this space. We knew a lot about neural engineering broadly, retinal prostheses specifically. We were well connected in that. We had access to the resources to pursue it. But I think the reality is that there are a lot of interesting ideas out there. And the thing that motivated us to do this acquisition were the clinical trial results that the patient video spoke for themselves, and as well as some insights on how we could run with it and take it further, and being in a position to do that. But the there's tons of interesting ideas out there, and I think it's not that how do you figure out which of them will work? It's that probably many of them can work, and that's just not how structurally a lot of this field is not how it views the world. I think a way in which we are a very unusual company for the medical device industry is medical devices and biotech broadly. Often you see companies that are kind of single purpose asset companies. They are things that are spun out of a bigger company or out of academia with a specific piece of IP that they're going to commercialize with some professional team that's done it 15 times before with some known playbook of investors. And it's like they work backwards from the regulatory strategy and the reimbursement analysis and all that. And then at the end, they get like version one to market, and then it's been 10 years and everybody is super tired, and version two never even ships. And I think bringing just the technology company playbook and mindset to this industry um, creates a lot of possibilities that are otherwise not um, like that are otherwise missed. Like I think a lot of these things can work, but you have to. It's versions 4, 5, and 6 of a lot of technologies that become really compelling, and the structurally, the way that a lot of this field is funded and the way that the experienced executive teams view it and how the regulators view it, they never get to versions 4, 5, and 6. They ship version 1 and maybe version 2, and then it disappears into Roche, or it gets um, it because like the technology now, the results are incredible, but it's still very far away from high resolution color vision, and I think we want to get there. And how do you keep up a high pace of iteration? And this is challenging. Like you can learn something every week in mice. You can learn something every 3 months in primates. You can learn one thing every 5 to 7 years in humans. And the and this is a very difficult problem. It's not enough just to say like, oh, this is the FDA's fault, or oh, there's some specific reform that could happen, because the structural issue there is that when the FDA approves 100 things that are really valuable, nobody thanks them. And then they do one thing that one patient gets injured, they get hauled before Congress and yelled at. And kind of in our infinite wisdom, our society has collectively turned our risk tolerance towards human subjects research down to like -5. And so that makes it very hard to iterate. Like you end up with these multi-year design freezes. Even now, like we know that this will work for a bunch of patients, and we are at least 16 months from being allowed to sell it to people. And this is entirely a regulatory delay, and they're rationally responding to their incentives. And so we think about like um, we have a large in-house animal program up to like some big animal species,
Mhm.
because that is where we can continue to learn things quickly, and that is very contrarian and it's very capital intensive
Yeah.
for the field.
So the reason I asked that is just because I'm curious about how you found this particular thing. Because neural engineering is a broad set of possibilities. There are
Yeah.
lots of senses. Like why choose this one? Like why start with sight? Was it a Was it more of like okay, we had a way to get there fast and move things quickly, or was it like there was
It's the
physical stuff with sight?
it's the intersection of these things. It's like it
Yeah.
both has to be attackable and it has to be a big unmet need. And so, um, we have ideas that I think will produce better cochlear implants. Like right now, cochlear implants basically work, but you can't take them to a concert. Like you can't really see live music with them in the same way. And so, absolutely, we have ideas that could build better cochlear implants, but it is a much more competitive market. It is less serious of an unmet medical need. It reimburses less well. Whereas in these blindness cases, these patients have nothing available. It is completely debilitating. We are incredibly visual animals. Um, Plus, we had a thesis about how to attack it technically. And when we started the company, there were a couple different approaches. And then, like, we weren't founded around a specific piece of IP. We had there was a little bit of an open question in our mind about whether. So again, there's 3 layers of cells in the retina. You've lost the first one. Do you drive the bipolar cells, or do you drive the ganglion cells? Those are a little bit different. The retinal ganglion cells are true neurons that have spikes. The bipolar cells do not. They have a kind of an analog release. Um, there's some more complexities around either of them, but really we narrowed in on you could either use an optogenetic gene therapy, where what you use is a viral vector. When people hear virus, they think of something that's going to like replicate itself endlessly. It these don't do that. They can't copy itself, but you can use kind of the machinery of a virus to put a new gene in a cell. And so you could deliver a gene that would make the cells sensitive to light, so we could use a projection system to activate them. Um, or you could use an electrical stimulator to activate them. There's kind of pros and cons of either. We we're working on both. As a company, we have what we understand to be some of the best of those proteins, those optogenic proteins, in specific categories of performance that we care about. Um, and we did a bunch of preclinical research on using optogenetics to restore vision. That got all the way up to uh, thinking about pulling the trigger on a first in human dosing, when the data came along for the electrical
Mm,
stimulator that this was really working in humans in a
cool.
way that I honestly didn't necessarily expect to work as well
Hmm.
as it did. And uh, that was just so much closer to commercialization that it's taken our focus for now.
How similar is this optical product to the actual implant that's going in people's heads?
So the other program that we have for biohybrid neural interfaces, this is a totally different thing.
Okay. The
Yeah.
like there's no
Com-
the because
well,
it's well the
yeah.
reason why I asked that is because I know that the implant is photo like the way they make the neurons fire
Yeah.
is light, right?
Yes. So
Okay.
optogenetics is a really powerful tool. So there's a couple different issues here. Or a couple different topics here. The first is uh, restoring vision. We commonly associate with light because it is a light sensitive
Right.
organ, but that is really different from using optogenetics as just a way to drive neurons specifically.
Hmm.
Like you can like digitally drive neurons selectively using optogenetics, totally separate from anything about sight.
Hmm.
Um, the in the biohybrid neural interface work that we do, where what we have is an electronic device that we grow up in a dish with heavily engineered neurons seated in it. And so in that device, there are micro LEDs and there's capacitive electrodes. And the cells basically grow on those and grow in these like trench structures where they're surrounded by LEDs and they're surrounded by electrodes. And what this allows us to do, and we make the cells light sensitive using by expressing an opsin protein in them, which is a light gated ion channel. So it's a protein that is expressed in the membrane of the neuron that when it is hit with light, it moves ions around to control the state of the cell.
Is this a natural part of like most neurons, or is this a specific thing you've grown?
They were it was discovered naturally, so I think it
Hmm.
was originally found in an algae.
Hmm. Wow.
And now they've been found throughout nature, but no, human neurons do not normally express
Hmm.
opsins,
Okay.
generally speaking.
Got it.
There are light sensitive proteins, obviously, that are expressed in rods and cones, but not in normal neurons in the brain. And so, what this allows us to do is we can activate the cells by turning on these micro LEDs to fire them. But then we can also, when they receive input from the brain, we can also detect spikes that they get from the brain using the electrodes. And so we can optically stimulate and electrically record. And this has the effect of really avoiding crosstalk, which is a system of like a function of
You can
any
receive
system
and send at the same
at
time,
the same
basically.
time.
Yeah.
Because if you electrically stimulate, then this will totally overwhelm your recording electrodes and takes a little while to recover. And then also just the physics of recording electrodes are just more challenging. Um, Electrodes are always, if you can do something with pure packets of energy, it is usually like what you want to do. Um, and so so that allows us to bidirectionally talk to these neurons that then when this is kind of seeded into the brain, those cells grow in, wire up, form new connections into the brain. And I think really you can have the high level intuition for what's happening here is like the brain is a bunch of neurons. What happens if we grow a bunch of neurons on the brain? It's just like more brain. And they grow in and they wire up as part of the brain. Except we've kept the neuron itself with us so that only the inputs and outputs grow out into the brain, allowing us to kind of have a part of the brain that is easy to digitally communicate with.
Yeah. And so obviously the implications of what you could do with that are
Yeah.
endless. I mean, you have input output to the brain basically at that
There's a
point.
ton of neuroscience to do here. I mean, it's like in some sense you could it's super you can imagine doing all kinds of things, but then actually using it to do specific
Yeah,
things is
exactly.
there's the
Unfathomably complex. Like
there's a lot of interesting neuroscience to do here. Have you seen the Avatar movies by James
I was gonna
Cameron?
ask this question. It's like the thing. It's like the
That
brain.
is actually how I conceptualize
That's
it.
what I'm
That's how I
talking
think
about.
about it. It's like it's a big externalized cranial nerve
Yeah,
with a USB-C port at the end.
exactly.
Yeah,
Exactly. Well, then this actually raises the important question of like how what on what stage of the roadmap is like we have this podcast by just connecting you in and then we
absolutely.
have our own conversation
Brain to
without
brain is
yeah.
one of the most interesting long-term
So cool.
implications of this. I hesitate to call it an application, but it's
Yeah,
definitely
sure.
an implication.
That's totally right.
And that, I mean, I have no idea what this is going to feel like,
Right, exactly.
but
You're just sensing what's happening in other
but I am
brain.
pretty confident that it is like less than a decade before we find out in humans.
That's so crazy. I mean, so okay, I have so many questions about this. I think the operative question first, before we go into like crazy sci-fi land, is like what are you building towards in the short term? Like, are there particular use cases of this thing? Do you want to use it to control external devices? Do you want to like go into particular, like be able to stimulate areas of the brain that have atrophied or something? Like, what's the immediate or like nearer term stuff before we get to the crazy sci-fi?
Yeah, so there's two ways to answer this. The first is we've architected the company so that we aren't actually really dependent on revenue from commercializing this in the near term. Visual prosthesis, like I hesitate to do cure blindness, but like as you make progress with curing blindness, that is that's a multi-billion-dollar a year cash flow stream. And so to the degree that we can have the space to do some of this research in things where we can iterate more quickly, because as soon as you go to humans, again, everything slows down. And this is too early stage to say we're going to design freeze these cells and we're going to design freeze this device for the next 5 years. And so I think some of the next major milestones are going to be in animal models. But with that said, you do need to think about what do you use this for in humans, because otherwise, really, it's just like what are you doing for? And there's a couple ways to answer that. The first is ultimately so BCI has become kind of synonymous with motor decoding, like controlling like keyboard and mouse signals, but that is the low end of a spectrum of communication, and we think of them as really good communication interfaces. And so you expect to be able to do things like keyboard and mouse control. We expect to be able to do things like speech decoding, but up to communicating more complex cognitive objects. Like imagine if you can hear, like, imagine a sound and you can copy it to a computer. Um, and that is definitely research, but that's kind of the direction that goes. The second is with the cell component, to some degree, you're on a spectrum with regenerative medicine, and there are applications in things like stroke or even degenerative disease that you might be able to think about attacking that really don't make sense with conventional electrodes, where if you've stroked out a part of the brain, there's nothing to record from, then there's nothing you're doing with electrodes there. Whereas if we're adding engrafting in big groups of cells into like a void created by an area of atrophy, then it might be possible to think about, again, like we don't think of ourselves so much as a brain computer interface company as interested in neural engineering, which goes beyond just IO channels and more about how do you think about like what is the architecture of the brain? What are the parts that fit together? I mean, it's crazy that really we don't have great answers to this for, I mean, there's a very long list of true facts that neuroscience has learned about the brain, but adding those up to a coherent whole picture is still often very challenging.
Totally. So on the input output side, like here's another movie comp. It's sort of like Arrival. You seen that movie? So like aliens land on Earth and they're trying to communicate with them, and they create this whole language between them.
Yeah.
That's like, okay, we start off and we can do 1, 2, 3, like that. We can show one dot and then show two dots, and we can figure out how to decode those things. And then eventually, towards like the end, you can have real sentences. You can really communicate back and forth. So like, is that like a fair model about like the input output challenge? Is like coming up with enough symbols that mean enough different things that you can really understand what's happening and communicate with the brain back and forth?
Yeah. I mean, from in terms of actually doing any of this in an implanted context, I think it's really early in that research. But the one of the weirder or one of the things that's been, I think, pretty striking is the representations that you see in the big AI models look a lot like the representations that you see in the brain. And this, I mean, this is not, I don't think I'm saying something that I think people in either of these fields are like, gonna be like, oh, like I haven't thought of that before. It's like, it's been striking the degree that this has happened. And the, um, but there's kind of an obvious question of why and what does this tell us? There's an idea floating around called the Platonic representation hypothesis, which is that both kind of all of these sufficiently powerful learning methods, it doesn't, irrespective of what objective or whether it's gradient descent or it's something else, they're all kind of grabbing onto the same underlying data manifold of the world, and this tends to produce the same kind of Platonic representations. And that's like one idea that
That there is a true structure of the world, and we're just
that you
finding
can
different
kind of
ways to
efficiently
approximate it.
learn, yeah.
Yeah.
And so if that's true, and then there's been a series of really interesting papers showing that you can use deep learning methods to learn um, kind of decoding models that actually can be conserved across individual animals, across individual subjects. Like obviously, the specific neurons, the specific way they're wired up, like the microstructure, of that very different. Like, and when you put an electrode in the brain, you're going to get some random neuron in that area of cortex, and it's going to have some that like the specific tuning of those cells will be totally different, but the general thing it encodes for will be conserved. So to make this concrete, in primary motor cortex, which is a strip kind of at the top of the brain, which is only a couple of synapses from the muscles, those cells, when you record from them, often, and the tuning that you see depends on the behavioral question that you're asking, but you'll often see things that kind of correspond to joint torques. So as you move around, the cells will fire proportional to how much force is being exerted through a joint. And that makes sense because this is controlling muscles. And so the output to that is going to encode joint torques. And so there's a representation of the body surface of like this manifold of the body and all of the degrees of freedom on that. And so even though the specific cells that you're recording from and the specific weights will vary animal to animal, you're still going to get this manifold of like body representation and coding joint torques, which in the body plan is conserved. And so that overall structure will be the same. Um, or if you record from other areas of the brain that correspond to things like they have these concept spaces. Like, have you heard of like the Jennifer Aniston neuron?
Oh yeah, sure.
Yeah.
Yeah, yeah,
So
yeah.
it's
Or like the
known.
grandmother neuron or something. I forget what the
Yeah.
yeah.
So there's like parts of the brain that if you record from and then you show subjects pictures, there's some that'll be very selective for things like a specific person or like the concept of zebras or and you can show them like different images that are more or less, and that neuron will fire proportionally more or less depending on how close it is to like the thing. And the specific neurons will change, but those manifolds are conserved. And so then people have been using these deep learning models to learn ways to take spike trains from one subject and kind of convert it to a spike train for another subject. And so back to the original question you were asking about Arrival and do we need to learn these translations? It looks like you can use the tools of deep learning to really get more that translates across brains than I necessarily would have thought, and certainly what we were thinking about 10 years ago. And there will always be some adaptation, and the adaptation will be both in the model and in the brain. The brain is very plastic under feedback, but my guess is that you'll be able to learn that, like those mappings will be able to be learned.
Yeah. And so that's why it's so hard to design for this, it seems, because so first of all, the subject that you're trying to learn about is still not perfectly understood. This is what you were talking about. It's like we understand bits and pieces. We know that this is the motor cortex. We know that's the sensory cortex, whatever. But like on the whole, we don't know what consciousness is really. Like, I don't.
Don't even,
I,
I mean,
like we,
we could talk for hours. I have so, yeah.
I would love
Co-
to hear you
This
talk for
is, I
hours
mean,
about
ultimately, I'd
consciousness,
say
but
like if there's like one ultimate goal, it's to understand the hard problem, which is like, don't even, we're not there yet. We'll
yeah,
worry about that
totally.
later.
But like, so, but like, that's like such a hard thing to engineer around then, because it's like we don't actually even know because normally in
Yeah.
engineering it's pretty like deterministic, right? It's like ones and zeros, and we can run every iteration of this software and we can prove that it is, going to always result in the thing that we want it to. Like that seems like totally
Yeah.
impossible here. It seems like it's more probabilistic as like neural engineering
Well,
than computer engineering.
Well, one answer is this is a reason I really like the retina, which
Yeah.
is as close to a feed-forward structure as you get in the brain. You go from photoreceptors to bipolars to ganglion cells into the brain. And a lot of what makes dealing with the brain really challenging is it's so recurrent. There's constant modulatory feedback. I know there's interest in driving vision by stimulating visual cortex, which is in the back of the head. So once you go out of the optic nerve, the next stop is a structure in the thalamus called the lateral geniculate nucleus, and from there out to primary visual cortex. But a problem with driving primary visual cortex, even just a couple of synapses later, is it is getting tons of modulatory feedback from later parts of the brain. And so like the representations that you get in V1 depend on things like attentional state, depends on if you're moving or standing still or if you're tracking an object. And so to know how to stimulate V1, you need to know what a lot of the rest of the brain is doing because there's so much modulatory feedback. And so there's, but with that said, it's also, it's very stable. It's a very homeostatic system. And so you the failure modes that you get on these things, like there's a lot of self-balancing going on, and so you don't need to be that right, like in the sense that it's stable. um there's some failure modes like getting like eliciting seizures, which is a failure of the stability. Those are pretty, like, you don't get those with anymore. But as long as you're in the region of stability, as long as you're relatively close, the brain will figure it out. Like the brain will learn to you. If you give the brain information, it will extract the meaning. And so that is a way in which the brain is amazing. Um, and uh, like I said, like the list of true facts that neuroscience has learned about the brain is very long. We sometimes have a hard time assembling that into like a totality picture, but all of
You can
those
do enough. You
individually,
can you
there's
can work
stuff
with
to
it. Yeah.
do. And yeah, and one of the things that we're doing at Science is making our kind of tech stack available to academics and others to really hopefully enable other experiments, because there's so much neuroscience to get done here, and to the degree that we can get these tools into the hands of a much larger global community. I think like Bill Joy of Sun and Kleiner always used to say, there's way more smart people outside your company than within it. And so we're hoping that to the degree that we can help build tools that then others will use to do more science, we see ourselves as a small part in that. And we're hopefully getting to a point where we're starting to ask more sophisticated, more complicated questions in these things. Um,
Very cool. No, I'm curious about. So, would you say that the near term goals for the interface are like, do you like, is it like a bandwidth thing? Like, are you just trying to get higher bandwidth in and out? Is it an accuracy thing? Like, what are
no,
the limitations of
yeah,
it now that you're trying to overcome?
I wouldn't think of it as a bandwidth thing. I think
Mm,
people often say, oh, with BCIs, you want to increase the IO and then you can like think
yeah,
faster.
sure.
There's uh, neuroscientist Marcus Meister who is, I think, gotten like really, like twerked on this recently, because people have been talking about this. And if you kind of look through the brain, there seems to be this like speed limit about 10 bits per second. Um, and I started calling it like the Meister rate. The People with eidetic memory, if you like take them on a helicopter ride over Manhattan and then you
Yeah,
ask them to draw it later,
draw the whole
and then
thing.
you
Yeah,
look
yeah.
at the detail that they actually capture, it is like semantically downsampled to about 10 bits per second.
Really fascinating.
If you um, there seems to be in our cognitive process, there's this fundamental limit around that. Now, that's not to say that it couldn't be expanded upon with some broader sense of neural engineering that we are not at yet, but um, 10 bits per second is still a lot. Like 2 to the 10, pick one from 2 to the 10 every second is like a pretty big space. And so I don't think that is actually that limiting. Imagine writing an essay, and as you're writing this, you're having thoughts that are occurring, and the rate at which you can generate the essay and you can receive information from like a prompt and generate something about it, that doesn't necessarily vary a ton with depth of education. I mean, there's like certainly some education required to be a good essayist, but how specialized you are in a field, like if someone knows a lot about science or someone is like a race car driver or someone is a painter, their ability to like they could be perfectly fluent at writing well-written essays, but the amount of information that they have available might be totally different, and the information that they can call upon might be totally different. And so how can you put these like pre-trained representations in parallel, like in how can you kind of add cortical areas that have access to these representations that allow you to do these transformations that allow you to make this available to the processing? um, and so we think of it more as like adding or removing c- cortical areas rather than changing the like maybe a way to think about it is like downloading knowledge.
Yeah, like
So
I
like
know
I
kung
know
fu.
kung fu,
Yeah.
right? That's not like suddenly knowing kung fu doesn't break the 10 bit per second Meister rate,
Hmm.
but it gives you access to these
Sure.
representations that you didn't have before.
Interesting. So in theory, you could, if you have this input output thing, if you have the functionality to interface deeply within the brain, then you could learn new skills, or you could
That's the
like
hope.
download a
I want
skill.
to be
Yeah.
clear that we have not
Sure.
definitively shown this yet,
Yeah.
but I think we
Cool.
have reasons to suspect that that will be possible.
So okay, I will put one quarter into the consciousness machine and ask you the question about like how does that change you, or like how does that change the person? Because I don't know, the thing that keeps coming back to my mind is like the ship of Theseus or whatever, or Thesis, I don't even know what, but the ship of Theseus. It's like if you change enough things about the thing, is it still the same? Like if we swap out
Yeah.
my motor cortex for a an artificial one, what happens? I don't know.
Yeah, so there's when I catch up with like the friends that are my friends that are interested in these things, there's a question that always like completely evenly divides the table,
Hmm.
which is a destructively scant upload you? Um,
Hmm.
there's
Yeah, if you're doing teleportation and you just like
yeah,
evaporate the entire first body, but reconstructed
exactly.
cell for cell, and the other one is it still you?
Yeah, so these reduces to three what we call the transporter problems,
Yes.
and I have to cite like Derek Parfit, and there's a bunch of people that have written about this, but I think it's like the they really reduce to right like if you the first one is just if you get a transporter that totally atomizes you and then reconstructs you on the other side with new elements locally sourced, is
Mhm.
this you?
Oh, yeah,
If
sure.
it deconstructs you but transmits the matter and reconstructs it, is it you?
Interesting.
um and the where I land on this, and if I get into arguments with friends, I think that I have friends that adamantly insist that is absolutely me. The thing in the computer like does not bother me. Like that's like it has my and it has my agency, has my memories. This is continuity. This is me. I don't buy that. From my perspective, the thing that matters is continuity. And so the transporter, if it created, so the situation you don't want to get in is that it doesn't destroy you, like take one down before it creates the other, because now there's two of you, and it's like what happens now. And I think actually what you really want is for at least some brief moment there to be a connection that gives you phenomenal binding through the interface. So
Oh,
there's a
then we have a
to
chance to avatar
continuity,
first. Yeah.
and then you can drop the other side. But you
Okay.
need that continuity. And I think
That
that
makes sense.
metaphysically where this breaks down is some people, their brain has just made a choice that they can't unsee, that the thing that matters is my agency and my values and my memories and continuity of those, versus accepting drift in personality and values and agency, but getting continuity of phenomenal experience. And I fall in that latter camp. And so that's one of the reasons I think that the brain to brain connections are so interesting. But um, how you get phenomenal binding over the network is like who knows? We're not there yet, but I I expect that it'll be possible. Um,
Well, the
and
thing that always messed me up about that train of thought, just to poke the bear a little bit on this, I think like there isn't really a difference for me. Like imagine that I just started existing in that moment. Like I still have all the memories of all the previous conversation that we had and like what
totally,
I ate for dinner or
you go
whatever,
to
like all
sleep.
that.
You
But
go to sleep at night. You wake
yeah,
up in the next day. I think like these
how
are
do
the
you know you?
responses
Yeah,
of the other side,
yeah, exactly.
and ultimately it's tough to respond to. And all I can say is like, well, if I was like diagnosed with stage 4 cancer, I just don't know that like another copy of me is going to make me feel better. It feels like
Yeah,
one of
sure.
my friends. Like I'm glad that my friends will live on without me, but that doesn't necessarily. I don't really find that inherently comforting.
Yeah,
And it's true that
but if
like
you could upload download, then it'd
going
be
to sleep is an inconsistency here, and our brains just like it's like these are all quirks of what our brains are doing. I think it's also possible that to just have the brain not be concerned with longevity, and feel as fine about being diagnosed with stage 4 cancer as it feels about like going to sleep, as long as like there's software that will carry on your kind of your agency.
You're building a neural engineering company. It
Yeah.
seems like a new category to me. Like it's not a thing that I've heard other people talk about in the past. Sure, there's other BCIs. Sure, there's other brain type startups or whatever, but it's a new category. So do you find it hard to find people who can plug into this? Do they have to have a lot of external knowledge or like past understanding of the brain to be really helpful to you, or can you find just a software engineer and teach them the ropes?
Well, I mean, the field that we come out of like systems neuroscience and all like that isn't small. So there's like a big community. And in some sense, it is relatively easy to hire because there's not that many places where you can do this stuff. And so it's like it's sometimes easier to do a hard thing than an easy thing because like people want to help you. And the people that I think it is often more fun, like getting to do this at this scale with these resources is way more interesting than writing grant applications every 4 months and like dealing with the uh it's just that the pros and cons are totally different. And I think when now that the corollary of that is we need a closed economic cycle, it is very important to have a real business in there, which is why the visual prosthesis is so important. We have some other um I think our mandate is a little broader even than just neural engineering. We can pursue really high conviction innovation in medical devices, and there's one other area where we have high conviction, but it's an earlier stage, smaller project, which ultimately has tie-ins to neural engineering, but is a little distinct. Um, and it
Also
but
more peripheral. Is it like I would call the eye like peripheral to the brain implant. So is it also sort of a more peripheral thing, or is it?
No, it's a little bit of a different category, but it builds on the same foundational infrastructure. And I think people often there's like if there's a misconception that people have about this stuff, it's that it moves forward by big scientific insights, and often it moves forward by infrastructure. Like there's the vertical integration is essential. We do everything from integrated circuit design to MEMS fab to animal surgery to cell culture to protein engineering. And if you are at housing for monkey capacity, then it is like not really easy. You can't like just add one more. It's like this is a possibly like a multi-year permitting and construction project. And so projects like this are really limited by infrastructure as much as they are by good ideas. Um, and kind of by extension capital, but not just capital. It's like knowing, I think I've learned way more than I ever wanted to know about and like building um like quarantine facilities,
Hmm.
um,
Wow.
and just like operating or like on the fab side,
That's like to
there's
prevent the Planet of the Apes scenario where
yeah.
it's like,
Well, I mean, no, I mean, you're in practice, you don't have real concerns about that. It's
okay,
just you always, as part
sure.
of like the routine, you always go through a routine process. But on like the fab side, we're working with very serious hazmats that um, require real skilled people and real infrastructure and real operations. And I mean, similar to space flight, um, you you can't really do that like virtualized through a laptop. It's a very atoms thing, and the and a way in which I think academia often is not as good at these very interdisciplinary projects is they tend to pull together deep experts that are very specialized from different parts. Like when they think about priding themselves in interdisciplinary research, what this means is they'll pull together kind of people that are very narrow but very deep and get them to collaborate. And in my experience, it usually works better if you have people that can hold more of the problem in their head and are very individually broad, and then you can augment them with deep expertise in whatever specific area. But the more that you can compress it, like the more parts you can delete, the more you can make the system simple. The fewer interfaces you have, the better. Like the more our devices approximate towards like a single block of covalently bonded matter, the higher performance, the lower energy, the smaller, the better. Like they'll be. Um, and this is a very different way of viewing the world. But I think when when stuff starts really working industrially, the types of resources that industry can muster are enormous relative to really anything else. And like you saw this in aerospace, you saw this in computer science. Like it used to be that the smartest computer scientists were at CMU and MIT and Stanford, and now There's obviously still many there, but there's also a lot at Google and Meta and OpenAI, and aerospace. Like in the 50s and 60s, if you wanted to work on rocket engines, you were at a aerospace engineering department at a university, and now they're at SpaceX and Blue Origin and other places. And so it's kind of, I think, a fair question to ask. Like, academia stayed very dominant in the life sciences. Why? And I think a key part of that answer is like humanity is not very good at it yet. We just aren't really good at drug discovery. And um, as this stuff starts to shift, as the thing that BCI and neural engineering need to have happen is there needs to be a company that's making a couple $100 million a year sustainably, and I think that will lead to an enormous, like an enormous revolution. But and I think we're getting close to that. I think I think that the retinal prostheses can do that, but um, there's no company that's doing that right now. But that is, I think, going to lead to a real overhaul in how a lot of this stuff is done.
And you're trying to build those resources to help people as like go other companies to do the same sort of thing.
Yeah, absolutely. I mean, we make a lot of the tools that we use internally. Our chips are so there's the data acquisition systems that people often use to do these, like even low channel count neural recording is like a cart costs $100,000. It's like a big rack mount system. Those are available from a handful of different companies. We we've shrunk it into basically a glorified Qualcomm smartphone circa 2021. It connects to Wi-Fi, so we call it sci-fi. And we have a couple different versions of that coming soon. And yeah, we have a kind of an explicit goal of getting those into the hands of as many researchers as we can, in the hopes that we can help them do more ambitious research. Because they certainly have the ideas. Like, we are not the smartest people out there, but it's just it is it is uneconomic to do that. Like, that couldn't be the heart of the company, because there is not enough revenue to be made from selling these devices to academic researchers, even though they are such an important part of pushing the knowledge frontier forward to enable these products and enable these impacts for patients. And we get to do that because it can be paid for by our work in blindness.
How similar is that to what you were doing way before, like with transcriptic?
It I mean, it's similar in that it's tools, but I think one of the lessons I learned really clearly at Transcriptic is, like, don't try and sell scientific tools to academics.
Sure.
Um, they simply like, Transcriptic ended up most of its customers ended up being big drug companies. Like, we had did a bunch of work for Lilly and others that I don't know if I can mention. Um, but when if an academic lab had the opportunity of complicating their experimental protocol significantly, but taking 25% off their per sample cost, they would do that every time. And that just, like, was not really, but again, like, life sciences is really hard. Like, humanity is just not good at it. Biology does not happen in the milliliter volumes that you're doing it in these plates. Biology happens in nanoliter and picoliter volumes, and we're not, this is very, very challenging still now. Certainly was 10 years ago when I was doing transcriptic. And so we ended up mostly serving big drug companies who were happy to pay, like, more per sample, simplify their protocol. Could do high throughput screening, and ultimately, I mean, the year that I left, it did over 10 million in revenue. Um, Ultimately, did not really have the impact I think we wished we would have had on the industry. Um, but I think there were lots of lessons that I took from there that we have that we've adopted at Science. Like, for example, our commercial foundry, we're doing that very differently than how I thought about things at Transcriptic, but I think we still have a desire to make these things widely available and get tools in the hands of lots of smart people.
Awesome. Well, thank you so much for coming on the show. This is really
Yeah,
fun. I
thanks for
think
having me.
I would love to end this off with you asking the audience if they could all read one book. What book do you think they all should read?
I'm gonna take a cop out on that one a little bit, and instead of a book to read, a TV show that they should watch, which is criminally underrated and basically nobody has heard about it. But there's a show called Pantheon,
Pantheon.
which you haven't heard about it.
No,
See, nobody's
I've
heard of
not.
this thing.
No.
So it was made by AMC Plus. In around 2020. It's adult animation, and for whatever reason, they just, like, didn't market it. And then they made 2 seasons. They canceled it before the second season got released. The first season is now on Netflix. The second season is you have to surf the high seas of the internet to find. Highly recommended. Um, there's some problematic metaphysics in the show, but it's probably one of the best depictions of how I think the next decade might go in many ways that I've ever seen in fiction. It's based on a series of short stories by Ken Liu, who's best known as the English language translator of the Three Body
Yeah,
Problem
yeah.
trilogy. Um, I highly recommend it, and
Very
I think
cool.
more people might watch a TV show than read a book.
Hell yeah.
But I would love for more people to know that story.
Awesome. Thank you so much. This is great.
Cool. Thanks for having me.